simple animals. Sponges, like all other animals, exhibit multicellularity, meaning that there's more than one cell. Our major difference between sponges and the other animals that we see in the animal kingdom is that sponges have no true tissues. Remember, tissues form from the ectoderm, the endoderm, and the mesoderm. Given the fact that sponges do not develop endoderm or mesoderm, they don't develop tissues. This doesn't mean that they're not animals. Sponges are still heterotrophic, meaning that they still get their food from outside sources. They're incapable of making their own food. They are eukaryotic cells. And as we said before, they exhibit multicellularity. If we look at a sponge, some of the important things to notice, some of the important anatomy, is the oslium. This is a large opening in the top of the sponge that allows water and waste materials to flow out of. The oslium is the exit. For things to enter the sponge, they would enter through the pores. And these pores that are found along the cell, or along the sponge, are what give the sponge's phylum Porphyria its name. The holdfast allows the sponge to fasten to rocks or the bottom of the floor or the bottom of the ocean. If we look at a cross section of the sponge, these collar cells are what allow water to be drawn in through the pores. These flagella on the collar cells whip back and forth, pushing the water out of the oslium, allowing water to then be pulled in and enter in through the pores. These T-shaped cells are our spicules. They're the support structure of the sponge. These spicules can be made of calcium carbonate, can be made of silica, or can be made of spongin. And we're actually going to use the type of spicule, the composition of the spicule, to classify the sponges a little bit later. These amoeba-like archaeocytes are involved in digestion. That's where digestion occurs within a cell, and the production of eggs and sperm. Sponges lack a nervous system because they lack tissues. However, the external epithelial-like cells can respond to touch and other chemical signals by closing the pores and allowing water to stop flowing in. Sponges reproduce both sexually and asexually. Asexual reproduction occurs by budding. Here we can see the life cycle of a sponge when sexual reproduction is involved. Adult sponges produce both eggs and sperm, making them a hermaphrodite. In many species, the same individuals produce both eggs and sperm. However, they don't produce it at the same time. This is what prevents self-fertilization. Sperm is released into the water through the oslium, where it then enters the female sponge through a pore. So I guess what we should see here is these sperms entering through the pores, and fertilization takes place in the mesophyll. These sperm are trapped by the collar cells, and the trapped sperm are delivered to the eggs inside the female body, where the fertilization takes place. The resulting zygote will turn into a larva and escape through the oslium. Unlike the adult, the larva is motile. It's free swimming. It's covered with cilia that propel it through the water, and as the larva grows and develops, it becomes more similar to an adult sponge, thereby losing its ability to swim. The phylum Porifera is made up of 3 classes, and contains between 5,000 and 10,000 members. Our 3 classes are separated by the type of support system that they have. Our first class has calcium carbonate spicule skeleton. And we can kind of see the similarities in the beginning part of their names. These sponges are small, somewhere between an inch to 12 inches. Most of them are colorless, like our sponge here, but can exhibit bright yellow, green, brown, orange, red forms. These sponges form tubes, like we can see here, and these tubes can be interconnected, clustered together in a massive irregular shape. This makes the sponge asymmetrical. It's sessile, meaning that it doesn't move, and is a filter feeder, feeding on organic matter and bacteria that enter through the pore and, after being digested, waste leaving through the oslium. Our second class of sponges, Demispongiae, can have a spicule skeleton made of silica or spongin, or both. The size varies, the color, texture, and shape varies, and even the type of symmetry. Some of these sponges are asymmetrical, others exhibit radial symmetry. They are sessile, they still are filter feeders, but they can be found in worldwide oceans, and some are specific to depths in marine or ocean water, while some can be also found in fresh water. Our last class is a silica-based sponge. These glass sponges have a silica spicule skeleton. These glass sponges can vary from 2 inches to 5 feet in length and width, and they tend to be more vase-shaped. If you think about a vase, it's made out of glass, so our glass sponges tend to be vase-shaped. We see a creamy yellow to white, sometimes appearing clean, more white, or dirty, more yellow, based on the particulate matter that can be found in the sponge. These sponges can also be radially symmetrical or asymmetrical. These sponges are deep water marine, somewhere between 30 and 22,000 feet. They like a cold, low light, and high levels of dissolved silica environment.